This is section 6.3 notes talking about protein synthesis. So why does our body need proteins? Well, they're used for basically everything the body does. So how it's built, how it behaves, for everything metabolic that our body does. So just some basic examples of some proteins for structure. One of them is called collagen. This is what's found in bones to make them rigid but flexible at the same time. There's also another one called keratin. This is in your hair and nails. And it kind of is what makes those skin cells harden and die. And they are a little bit collagen based too. So they're a little bit flexible, but at the same time, nice and hard and protecting like the tip of the nail and protecting your head. And then melanin is a pigment in skin that gives us our skin color. So another type of protein that's really important. A couple other ones we've already talked about: DNA polymerase. That's used for DNA replication to add our complementary base pairs on. And then there's another one called lipase, and that's ASE. These are both enzymes, but they are a type of protein. And lipase helps break down all the lipids we eat in our daily diets. So repeating kind of that central dogma that we already talked about, DNA makes RNA, and then RNA is used to make protein. So DNA into RNA is called transcription. This is happening in the nucleus because the DNA cannot leave the nucleus. So we basically unzip the double helix, and then RNA comes in, makes a copy of it, and then that's allowed to leave. The RNA then, once it leaves, it can go out into the cytoplasm, find a ribosome, and that's where we can actually make the protein, and that's called translation. This is where we start grabbing a bunch of amino acids. You link them together, and a whole bunch of amino acids linked together help build a protein. So let's start with transcription. This is in the nucleus, and we basically find a chromosome, and we find a certain gene that we want to copy. So we locate that gene on that chromosome for whatever proteins we need to build. Our enzyme called DNA helicase, just like back in replication, that comes along. It unwinds or unzips the DNA for us so that we can have access to those base pairs. And then another enzyme called RNA polymerase starts reading that one strand of the DNA and adding complementary RNA strands. Now remember, for complementary RNA, there's no thymine. So instead of thymine, we replace it with a U, which is uracil. So everything else stays the same, though we still have A's, C's, and G's. So just kind of keep that in mind as you're practicing your coding. So the mRNA, once we've copied it, it becomes what we say spliced. It's where we're actually going to take some pieces out of it that we don't need, and those pieces are called introns. So literally, if you want to remember it, you're going to leave them in the nucleus, okay? So they stay behind. They're not going to come with you. Exons are the ones we need to actually make the proteins. So those are the really important pieces of genetic information. We take those out. We basically splice them together, meaning we kind of hook those pieces together. And that's the piece then that leaves the nucleus and heads out into the cytoplasm. So we're leaving the nucleus with only the necessary pieces for the proteins we need to make. So practice transcribing this DNA code into mRNA, keeping in mind that there's no longer T. Anytime you would originally place the T, you have to put a U now. So go ahead, give this a try. Okay, so if you remember the rule, it should look something like this. So T still pairs with A, but A now has to pair with U. C's and G's stay the same. So go ahead and check your code, pause it if you need to. All right, and on to the next step, what we call translation. This is where that mRNA, so the messenger RNA that was copied, now heads out into the cytoplasm and finds a ribosome. It's going to dock onto that ribosome and kind of stay hooked up onto it. That ribosome is also going to allow the other type of RNA called tRNA to start matching up with the messenger strand. So we have a group of 3 bases on mRNA that's called a codon. And that codon is going to tell us what anticodon to bring in. And an anticodon is 3 complementary bases on tRNA. So basically, mRNA and tRNA are always going to match up complementary. So just like you practiced on the last page where the letters complement each other, that's what's occurring here. So it's kind of being read, though, in groups of 3 called codons. So tRNA is this picture in the lower right-hand corner. It literally kind of looks like a little lowercase t, and one side would be holding the anticodon, the complementary base pairs, and then the other side is holding an amino acid. And that amino acid is basically coming in based on what the mRNA code is telling us to bring. So each set of 3 letters codes for one specific type of amino acid. We're going to start linking them together and making an amino acid chain, and that's actually how you build a protein. There's a couple websites here with demonstrations. I know the links aren't interactive, so you'd have to pause it and type those in manually. That bottom one might be a little tricky. So tomorrow we are going to run through some of these on the board together so that you can actually see how this process works. Otherwise, try this first one out. It's a pretty good one. So to actually translate the code then, that codon, that group of 3 letters, is going to tell us what amino acid comes in. And you always start with something called a start codon. It's usually going to be AUG, that sequence of letters. And then our stop codon is going to tell us to quit making the protein. And it'll literally kind of be called a stop codon. So to read this chart, you kind of have to go with your first base letter. So like we'll just use AUG. So my first base is an A. So I come down here on the left. There's my A. My second letter up top is a U. So AU meet in the middle here. And find G, AUG, is methionine. That's my first amino acid, and it is actually a start codon. It tells us to start the process of making a protein. So I'd write, and if I just wanted to abbreviate it, I'd put MET. So what you need to do right now is use this chart. So first base on the left, second base up top, and then once you match them up, square by square, find the amino acid that it links with. So start with AUG, we just did that one. Now go on to ACA. Remember they're read in groups of 3. So if you coded it correctly, it should look something like this. So MET, THR, ARG, TYR, ALA, and stop would be our sequence for a protein. Now, obviously proteins are usually hundreds of sequences long, but just for the sake of practicing, we kept these kind of nice and short for you, just so you can get the practice without actually going through the pain of making a true long protein. And again, here's another practice website where you can get some practice making the proteins, but we are going to be running through this in class together and kind of doing a couple activities to really reinforce some of these ideas. So finishing step, those amino acids have to link together. So, and they're held together by a peptide bond, and it's called a polypeptide then, once we link one amino acid to the next. So, however, once you've linked them, it's still not a functioning protein. We have to fold that protein, package it up, and then ship it to wherever it's needed in the body to do its job. So those polypeptides are sent to the Golgi. That's where that comes into play. That's your kind of FedEx center. It's going to actually fold that protein, get it ready to ship out, and then using the ER, we can take it wherever we need to in the cell, or we can get it out of the cell and somewhere else in the body. So this picture down here is showing a hemoglobin protein that has been folded about 4 times. It's one of the most complex proteins that we have in our body, and the Golgi helped make that. And that's it for this section of notes.